very much for your kind introduction, and thank you very much for inviting me here. This will be lots of fun, I think. So what I'm going to talk about today is, first, I'm going to talk a little bit about the core that I'm at and what we do at Lund Stem Cell Center, but mainly I'm going to be focusing on using the non-cutting but cutting-edge gene editing technology, which is CRISPRa and CRISPRi. So I'm at the Lund Stem Cell Center at Lund University. We have a core that's called the Cell and Gene Therapy core, and we do gene edits. We work on IPS a lot, and we also do viral vector preparation, so AAV and lentivirus. And this is sort of an overview of our services. So we are an open access infrastructure. We are available for all academic customers, international and national, and it's a fee for service, and it's still quite subsidized because of our sponsorship from the Stem Cell Center. We reprogram IPS. We do quality controls of those IPS, but also of other IPS if you come to us with your cells. We make custom edits in iPSCs, so that means that we do any type of CRISPR in those that you would like. And then also we do CRISPR design for any other cell type that isn't IPS. And then we make the AAVs, and soon we are going to start with some mRNA production, and we're going to have an IPS course, and we might start with some IPS differentiation. So I would just like to talk about this a little bit. Of course, previous speakers have already mentioned this, but you know, iPSCs and CRISPR really is this magic duo. The combination of those two quite revolutionary tools have really even further revolutionized research and potentially moving things into cell therapy. So the importance here, of course, is that you can take human cells and patient cells. You know, some of them would have no mutations, but some of them have unknown etiology, and you turn them into iPSCs. You can then edit these cells if you want to, and then you can differentiate them into almost any cell in the body, which of course can be used to basic research, to do disease modeling. You can create really large data sets from cell model. Of course, these are not neurons from a brain, but you could have potentially like 100 skin samples from autistic patients, and then you can turn all of those into neurons, for example, and then you could compare those. And then eventually, of course, this could also be put back into the patient as a cell therapy. But today, I'm not going to talk that much about that. I'm actually going to talk about the other sort of non-cutting use of the CRISPR technology. So on the left side here, let me see if I can also do a pointer. Not so big. Okay. So what we can see here is the normal Cas9. So here is Cas9. It has two nuclease domains that cuts the DNA, and you have the guide RNA. So what you can do is that if you mutate both of these two domains, then you get what is called dead Cas9. The thing is, it doesn't cut DNA anymore, but it still binds to DNA in the same way that the normal Cas9 does. So it uses the nucleotide or the sequence similarity as well as the PAM sequence to bind to the DNA in a very specific position. Then you can fuse things to dead Cas9, such as a CRAB domain, which then turns this into an inhibitor. And if you put it in the right position, you then inhibit gene expression. If you fuse an activator, or many activators, as it often turns out, to the dead Cas9, then you get an activator. So then you put that a little bit upstream of the transcription start site, and then you can get activation of a gene that wasn't there before. There are lots of other things you can also fuse to dead Cas9, but I'm going to focus on these two today. So we've done, the most of the work has been done with CRISPRi. So here we use the system, like I said before. We normally design 3 guide RNAs that is just downstream of the transcription start site. So this is used with an algorithm from CRISPR-Pic, and it seems to work really, really well. We deliver this using a lentivirus, and this is a fantastic virus, I must say. It makes a great virus. It works really, really well. It's high titer, everything like that. This is from Agene. I will show you at the end. It has a UBC promoter. Then comes the dead Cas9, which is fused to GFP. And then we have, no, it's not fused actually, apologies, but it's CRAB is fused to the dead Cas9. And then in the same vector, we have the guide RNA. So the U6 promoter going the other direction, and then comes the guide RNA specific for where you want to go. So CRISPRi is really excellent, I must say. Often people compare it to RNAi or other types of silencing on the RNA level. And CRISPRi is more efficient. So here is just one study that has shown a little bit that if you try to knock down the same genes, the CRISPRi is more efficient than the RNAi. But what is also important is that it is much more specific. So these diagrams here, what you really want is to have, it's a little bit difficult, but what you want to have is your dots down here. You want to have reproducible on-target effects. So you have them here as well with the RNA. It's not that it doesn't work, it works, but you have a lot of off-targets, and you can see that in these graphs as well. So it's much more specific. So from that point of view, it is really much better to use CRISPRi than to use any of these other methods. So I'm going to talk about some data that we generated using this method. So the cell type that we wanted to really evaluate everything in is a forebrain neuroprogenitor cell. So they're iPS derived, but basically is that you start with iPS and then it takes 14 days and then you get a forebrain neuroprogenitor cell that you sort of have to harvest around that time. And we always did it like this. So we decided to go with 2 different guide RNAs, and then we had a non-targeting guide RNA, in this case against like Z, but you can also use actual nonsense targeting guide RNAs to make sure that there is no effect of the virus or the CRISPR machinery inside the cells. So what we did was that we added the virus to the iPSCs, and then we expanded them for like 7 to 10 days so that they, relaxed in their new setting. And then we started this protocol. And then at day 14, we would then look for immunohistochemistry to make sure that they actually did turn into forebrain neuroprogenitors. We did RNA sequencing and qRT-PCR to make sure that the knockdown is done. So that's sort of the general scheme. But before we go into some data, I want to also show here that CRISPRi efficiency, even though it is more efficient than small interfering RNAs and so on, it is both guide and dose dependent. So this is against Huntington. This is actually done in fibroblasts, so it works equally well there. But what you can quite clearly see is that we have the different guides work differently well. I mean, guide 1 is almost not knocking anything down at all, whereas guide 2 works very efficiently and guide 3 a little bit less. And then if you add more of the virus, we can see that it works a little bit better with all of them, some of them still not that well. So it is both guide and dose dependent, and this is part of this wonderful paper that was published by Carolina Pirsch. And it also holds for a long time. But this is important. So you should always try a few different guides because you can see they vary quite a lot. So then using the method that I showed before is that you start with zinc finger 558. Now you start with the iPS, and this time we wanted to knock down zinc finger 558. And what you can see here on the left-hand side is that when we, while they're still iPS, before we have started the differentiation protocol, we wanted to see if the knockdown was working. So we just took them as iPCs. And you can see here, this is where we have this excellent virus. 92% of the cells are actually green. So that's really wonderful. And we can see that we have a very sufficient, very robust knockdown of zinc finger 558 with using both guide 2 and guide 3. And then we differentiate them for 14 days. And we can see that after 14 days, we still have 93% efficiency. And what we also see is that the knockdown is very stable, still around only 1 to 2% as assessed by qPCR. And we will actually show later that this was actually just noise coming from the system. So this is just another example of another zinc fingers. I must say, zinc fingers work extremely well for this method. Some work maybe a little bit less well, but still works really well. But you have to maybe not always that you get an absolute reduction of all the mRNA. So this is just showing that it's very efficient and stable. Also, once you turn them into NPCs, the knockdown is still there. It is even so stable that we took the IPCs and also turned them into organoids, so cerebral organoids. And even after 4 months, we could see that the knockdown was still very, very efficient. So these are control cells. These are the ones that have a guide of some sort. And you can see that there is almost no expression of 558 here. And these cells are not sorted. So this really shows that there is a really very stable transgene that we get delivered into these cells. So it's a really, really good virus. And CRISPRi works for a really long time. The interesting thing, though, is actually that a lot of the time we get actually complete silencing. So this is an example here. So cut and run is done by Chris Dowse here at the Lund Stem Cell Center, and he's really an expert in this. So I will show his figures here. And basically what you have is like, this is the RNA sequencing. So here we have a control, and then here after CRISPRi, you can see that the RNA sequencing signal is completely disappeared. But not just that, we can also see that, of course, there's a repression mark. So this is H3K9 methylation, which normally is put over genes that are not active. Now, of course, we are depositing that by using CRAB fused onto Cas9, but you can see it's not here before and it is here after. But even more importantly is that the activation mark shown here as the H3K4 methylation, which is there, the peak is there, so the gene is going in that direction. The gene, this is here in the control, but it's actually disappeared after CRISPRi. So this means that this can also be a very efficient alternative to using a knockout. Of course, here if you want a complete knockout, you have to continue, so you have to use a lentivirus. But this can be extremely efficient, and you don't have to go in and cut in the IPCs or in your cells in general. And this can, of course, be both cost effective, but also does not create any additional damage to the cells by adding double-stranded breaks and so on. And here's another example from the paper that I was showing. Again, we have an activation mark here. This is in the paper, very strong activation peak at the control, but then after we added the guide RNAs, there is nothing left. So you can achieve complete silencing if you want to. The thing is, you might not want to, like that is also the thing, you might not want to have complete silencing. You might only want to have a reduction in the levels. So therefore you can titrate your virus so that you basically put on to the amount where you are happy with it. And if you only want 50%, you can go with that, or 80% or so on. So you can actually try to achieve different variations of how much you want to reduce from the cells. So when it comes to off targets, of course, CRISPRi has off targets, just like all the other Cas9, basically. We have seen them sometimes very specifically also with the cut and run, that it's definitely there, and it definitely deposits the repression mark. The way that we deal with it here, and I often recommend that people should do, is that you use 2 different guides. And then you combine the analysis of that. So basically, as I've shown here in this schematic, is that you have the control, and of course you do RNA-seq, and that becomes your control. Then you do guide 1, you sort them, you do RNA-seq, guide 2, sort them, do RNA-seq, and then you combine these 2, so they now become the CRISPRi. The way that this works is, of course, that off targets that are only present in one of the guides doesn't become significant, right? So therefore, you should only see effects that are present in both guides. So that should theoretically eliminate the vast majority of all off targets, because they're very unlikely to have the same off targets. Like, it really doesn't happen. So this is a really good way to combine it. And then so you don't look at guide 1 versus guide 2, you look at CRISPRi, and that is, then you should really eliminate the majority of the off targets. So that you get a really true effect. This is, of course, the case with a lot of CRISPR. Even when you do knockouts, when you do SNPs, there can be off targets coming from the guide RNA itself. And the best way to deal with that, if you're concerned about that, is to do the same experiment with another guide. But here it's easier because, of course, what we're doing here is that we're keeping the cells in bulk, even though they are iPSCs. We don't go clonal, because in the end, every time you go clonal, there are serious risks to the serious risk for the genomic integrity, and especially with iPSCs. There is also every single cell expansion can create all sorts of things. And of course, that takes time and is really time consuming. So with a lot of things, even with knockouts, depending on your experiments, you can go bulk. And so one should never forget that it's not always necessary to go for single cell. What is also nice is that CRISPRi can be multiplexed. Of course, we didn't find this. Other people have shown this before, but it's just nice to show that in our hands as well. 2 different genes. This hasn't been published yet, so I won't show the ones, but one of them is not zinc finger. And what you can see here is that even though these were combined at the same time, so we used one virus that had the dead Cas9 and the guide RNA versus gene 1, so this is the same virus as before. And then we had one virus that only contained a guide RNA versus gene 2. And then so these were done exactly at the same time. And we have very, very efficient knockdown of both genes here as well. So the benefits of using CRISPRi itself is that there are no DNA breaks. It is easier to multiplex because you don't have to worry about big translocations or deletions or things that can happen. You can actually dose the knockdown, so not the knockdown itself, but like you can decide how much you want it. It has a much faster readout, right? Like all you do is a qRT-PCR, and then you know that the mRNA is gone. Then you know that this is a knockout or a knockdown. You know exactly where you stand. The negative, I suppose, is that it involves lentiviral vectors, so you have to be able to use them. You have to have the money to produce them. Which doesn't have to be that expensive either. But yeah, exactly. So that is like the thing, you have to use viruses. Unless you want to do a transient knockdown, in which case you can just use plasmids. And that works really well as well, but then it's only transient. The reason to stay in bulk is that you don't need to go for clonal expansion or expensive sequencing. It is better for genetic stability for the whole population. The same, because we're using viruses, there is a transgene insertion point, of course, but they will insert in slightly different places. So here again, we have thousands of clones in the same thing. So any ill effects that has happened due to expansion, due to insertion, or also then silencing of the population of the transgene in the population should also be like sort of nullified by the fact that we have so many clones. And in a lot of ways, it's easier, right? Like it's faster, you don't have to go clonal expansion, so you save time and you save money. Right. So all of this is in a STAR protocol if you're interested exactly how to design, how to troubleshoot, you know how it is with STAR protocols. So these are the plasmids that I've been talking about. So this is the one, it's from Agene. It says in the paper as well exactly which ones it is. So this is with a GFP. It also exists as a pure myosin version, so you can use that to select instead. This one we are trying to make, but are getting some sort of problems with that. But what we also have that you can get from the core are guide RNA only viruses. So they're red or green and then contain guide RNA and the fluorophore. And we also have control plasmids that has non-targeting guide RNAs. So that's sort of it for CRISPRi. I think it's a super, super good tool. I think a lot of people go for knockout when you don't need to. It is absolutely not necessary that you use it for iPSCs. You can use it in lots of different cell types. So we used it in fibroblasts, for example. Fibroblasts that were then turned into neurons, and so ions induced neurons, and that stayed really well as well there as well. So I think as long as your cell type can handle the virus, it's a very good way. But of course, it varies from cell type to cell type, from everything varies from locus to locus. But I think it's worth testing actually in your cell type. And there are slightly other viruses and plasmids as well if this particular promoter doesn't work for you. But it's worth a go, I think, for a lot of things. CRISPRa works on almost the same principle, but is a little bit more difficult. So here you don't design the guide RNAs downstream of the transcription start site, but actually upstream, sort of around the promoter. Here is much more difficult to find the right place. People have had a tendency to use multiple guide RNAs because they say it works better. I think some recent data is showing that it doesn't work so much better. There isn't really an additive effect, but it makes it more likely for you to find one that works. But I will not keep that. I mean, I've only heard this from another presentation. So, but I think that sort of makes sense. Some of them work really well and others do not. So what you can add to CRISPRa is like you can have VP64. There are lots more like advanced versions, so VP 194, 192 maybe. They can also add VPR, so different types, many types of activator systems. They all work. I think it depends a little bit on what you want. Many of them involve several plasmids, so you have to make like 3 viruses. This one, where we tried here, which is the simplest one, which in the end was the one that worked the best for us, is 2 plasmids. So the whole dead Cas9 VP64 with a selector plus the guide RNA does not fit into a lentiviral vector. So the guide RNA has to go on the side. So then you have to always use 2. But that you can, of course, do and have used different colors. So when we wanted to try it, we started with a positive control that somebody else had done it already. So we used SOC17, which is actually not on in iPSCs. And this is stuff from this other paper to show that, you know, when you use this guide RNA, you get it to work. So we wanted to try that. So we tried theirs. So the system that we used is that we had our iPSCs, we then added our green VP64 dead Cas9 virus. This was not a good virus, so it was very low titer, so we got an extremely low percentage of cells. So we had to like expand those few, make a new sort of cell line, and then we added the guide RNA virus to that. And then we sorted the yellow cells, and then we did our testing on that. And so here you can see that this is a little bit more problematic. Now, I'm not saying that you can't use it, but you just have to be sort of prepared for this to be a little bit more difficult. There are many, many different plasmids, many different versions of all this out there, and some of them work better than others for sure, but just so you know. So what we could see here was that 10 days after the last transduction is that we got like 46% that were positive for both of them. So that was great, lots of cells to sort. And also we had some that were only green, so that also makes sense in a way. But we also had this really large population that was no longer green, right? So they were all green when we started, but the addition of another virus made it not green. So then the transgene was silenced. So this was a really large percentage. So it's something one have to make sure when you manipulate the cells again that you don't lose the transgene. But either way, we found that indeed we got upregulation of SOC17. And so here, of course, we cannot use a number here because it's actually off, but you can see that now it's definitely on. The CT values are very high now, so it's totally fine. And the same with zinc finger 248. So this is a zinc finger that is on in iPSCs, but not very much. So here we went from very low expression to higher expression. So we can see that it works here as well. Yeah, so that's basically what I have to say about CRISPR. I think it can really work though, and you really have to try more guides. You have to try different activators. So it's a little bit more of testing and efficiency testing before you start doing it, and be aware that these are huge viruses. And I think it's worth definitely for all of these things to do a selection before you actually do your final analysis. So I'm like going to finish a bit early, so I'm sorry about that. What I also would like to point out, now I haven't actually done this myself, but I have collaborators from other countries, and especially then Ras Torkovic here, that is from Helsinki, that have done CRISPR-A. We use CRISPR-A for iPSC reprogramming, so taking fibroblasts and turning them into iPSCs instead of using mRNA or Sendai virus, which is extremely costly. You can also use just plasmids, so like somal reprogramming, which also works and is less expensive, but is not as efficient as this particular method. So if you're interested in reprogramming your fibroblasts into iPSCs, if you don't have a good core facility around, I can do it for you. Then I would try looking into this as well, because that is a much cheaper version instead of using, for example, the Sendai virus that is very, very expensive. So that was just more another thing that CRISPRa can be used for. So I think that could be very nice. I would also like just to do some advertising for core facilities, either for you to use them or for your institute to establish them. We are not just extra hands, you know, like where you can save money, which is true. Like you do save money and you get extra hands if you don't have enough researchers to do your stuff. You can get us to do it. But what is also really important is that we are sort of the keepers of quality. So we try to maintain the expertise, always be involved, and keeping up to date with everything, having standard operating procedures, being the ones that say, no, it's not okay to do this without doing the karyotyping, you know, to be the ones that go, look, we have to do this. And I think that is important, especially if you're looking at a cost perspective, because it is expensive to work with CRISPR, it's expensive to work with iPSCs, but you have to do it right, because otherwise it's all for nothing, right? And I think there are some people are like, well, we can't all afford it, and it's terrible, but you don't want to do it halfway, because then find a collaborator, you know, because if you do it halfway, you might go, the reviewer will ask you for something that you didn't do. So it's hard and it's a tricky situation, and I do understand that. This is the team. So I have a very talented, very educated team of experts of various kinds. The director of the core is Anna Falk. And yeah, so you can reach us at Cell and Gene Therapy Core. There's also a website if you want to use our services or have any other questions. So yeah, with that, I am done. Thank you for having me.
Thank you, Pia. Again, a nice presentation, and we had a lot of. So actually, it's the third conference today, and again, it highlights the importance of having a core facility of CRISPR in each university or each center. So we should install a CRISPR engineering unit here in PT. Before going to the audience, I have actually two technical questions.
Yes.
So when we deal with the stem cells and for transfection, is it because it's also particular cells, etc.? So is it so we have to transfect the cell and then go to the organoid, or we first build the organoid somehow and go to the transfection? It's for student audience here, you know, to
yeah.
Also, so the second question, we have groups here that are working on mitochondria. So is it also the same protocol? Is it also the same protocols, difficulties? So this is actually the second question, and thank you.
Yes, so with the first question, I mean, obviously it's much easier to first just do your edit in the iPSCs and then turn them into organoids. Sometimes you don't want that because, you know, your gene, you want to really go, no, I don't want to investigate from here. I want to start at this particular point. There are various kinds of sort of inducing it, but it is a little bit complex, and often inducing the Cas9, if you use a virus, for example, early on, they are a bit leaky and so on. So it's difficult. So the best way to do it is to engineer your iPSCs so that they have Cas9 in it, and then it turns on when you add the doxycycline. But that's like two pretty big edits often, which cost a lot of money. The other way is, of course, to do it in the organoids. And I mean, some people just electroporate the organoids. With different types of,
It works?
It works. Yeah, I think so. I mean, I don't know if it works with adding, for example, RNP, but it certainly works for adding, you know, plasmids of various kinds. And since you can, you know, use Cas9 in plasmid form. The question there, of course, if you have a multi-tissue, which ones, like how, especially if you want to do knockouts, right? Like, how do you know that this particular cell is a knockout? And what about the other one? Because not even all the green cells are going to be knockouts. A lot of them are not going to be it, which is this mystery of CRISPR, right? That some of them, even after 3 weeks in culture, still hasn't cut. So that is difficult. So therefore, if you can, I would, recommend, or you would have to like sort of take the green cells, then like have multiples, right? And then you would have to take them and see what your like general knockdown efficiency is, or knockout efficiency, and then sort of work with that, unless you can stain. If you can stain, in the organoids, then you're problem solved. But it's and if you probably heard this from the pod, then is that, it's always like that. There are so many like levels of your readout, and that's something that people forget sometimes, that even though the initial design to making the knockout or something like that is not so complex, your readout can be super complex if you have a complex tissue, if you have a mouse, or if you have like organoids and so on. And people are starting now to get ways to electroporate the whole organoids and so on. So I mean, people are working on it for sure. I haven't done that myself. We don't differentiate at the moment, so we don't do that, but it will come for sure. But it's always, remember, the readout. Green doesn't necessarily mean done. And with the mitochondria, people can do that. I have no idea, but you can edit the mitochondrial DNA as well. I'm pretty sure there was a paper, but no, I don't.
But we will work on it.
Yeah, of course. I mean, I think that's the thing. People have shown that it's possible, so then you just have to follow their instructions.
Absolutely. Any question from the audience, maybe? Yes, please. Because so in which cases CRISPR-A is more suitable than knockout?
Pardon?
In which cases or in which circumstance CRISPR-A is more efficient or more recommended?
Oh, you mean CRISPRi?
Yeah.
Okay, yeah, I mean, that depends on the locus as well. We had some where we tried to make a knockout first, and we just got like 70% or something, and we wanted to use bulk. So we were like, no. Whereas the CRISPRi was super, super efficient. I would say that CRISPRi works more often than maybe a knockout. But sometimes you have so much, if you have an mRNA that is so efficient, like there's so much of it in the cell, even you putting on as much guide, like as much virus as you can, it might not be enough to sort of get rid of it. That's the thing. It might be, it's reduced, of course, significantly reduced, like 100-folds, but it's still there. So it depends also on your, so then you would have to use your readouts to check downstream targets and so on. But I think it works. And with a knockout, I mean, you can do bulk there as well. If you want to just do it on a sort of RNA level, for example. But you have to check, of course, how big it is. And if it's like around 50-60%, it's just not enough. You have to have much more. And it's more likely that you get a level that is acceptable with CRISPRi, I would say.
Absolutely. Other question? Yes? Two questions, even better, yeah. Attends, yeah.
You hear me?
Yes.
First question is about CRISPRi and CRISPRi. So basically, CRISPRi is the interference of CRISPR and CRISPRa, an activation of the gene technology. So I saw in the slide, but the same system. So what makes the difference, or what makes the
so what is basically the main difference between CRISPR-A and CRISPR-I?
Right, outcome or like, so outcome is of course that, one is activating and the other one's repressing, right? The way that it works is that well, basically you just deposit different things onto the genome. So the guide RNA like calls the guide the Cas9 over, basically, so it binds and then it deposits whatever it is that you have attached to it. So if that's a GFP, you can see where it is on the genome. If it's a CRAB domain, which is a repressing domain, so any method that puts CRAB or puts this CRAB protein or CRAB domain onto DNA will repress sort of that region. Now, of course, it has to be a region where you normally instigate transcription and so on. But that's sort of what happens. I mean, the truth is also sometimes it's much easier to repress you can actually put dead Cas9 without a CRAB domain on it, and it often represses still. Whereas activation is much harder, right? Because there are lots of things you have to get rid of. And so it's a much harder process to do it. But in the beginning, like the start is the same. Something that is fused to dead Cas9, you use it as a sequence similarity to get it there. But yeah, outcomes are very difficult. And the difficulties of them are very different as well, even though the starting point is very similar.
Thank you.
Second question, yes.
The bulk and the CRISPR.
Bulk and CRISPR, yes.
Is it? I didn't hear before about that, but is it also an interference modification?
So basically, bulk. What it is? Well, was it bulk when we said bulk?
Oh, it's just that you take the cells and then we sort them, right? Because they're green. So we just take all the green cells, but then we keep them like that. So we don't make a cell line. We just make like a bulk line. So they haven't been, so we have a cell line that is coming from 1,000 different clones or more, like 100,000 that we started with at first. So that makes that, it just means that you don't have to do the clonal expansion, basically.
Yeah.
Which you do with a lot of cell types if they can't handle the clonal expansion, right?
Absolutely. It's okay. Maybe any other question?
I think there's one in the chat also.
Hi, Mohammed. I have one. Sorry, I'm online.
Hi.
Hi, yeah.
Pia. How are you? Thank you for a wonderful talk.
Thank you.
I have two questions. The first is, how do you account for off-target effects that result from being on target? And what I mean by that is like higher order impact on chromatin architecture or things like condensates, for example, because that would be really hard to pick up, right, in that two-guide method that you described.
Yeah. Yes.
So that's the first question.
Yeah, it's true. I don't know how to look for that. I mean, in the end, that should also lead to eventually to transcript. Like, for example, we used RNA-seq as our readout, right? But you can use whatever you want. But like then you would eventually higher order changes should lead to changes probably in gene expression, and then it's probably unlikely that two different guides will give exactly the same changes in the chromatin rearrangement, right? So then they should still somehow be separated from each other. But of course, much more complex, and don't know of the likelihood of them creating the same thing there, because maybe it's just, but yeah. And also that there's different chromatin marks. Yeah, I don't know how that affects the whole bigger order of things.
Super complicated. And then my second question is around the bulk method. How do you then deconvolve later on the impact of this bulk edited or heterogeneous population, essentially, right? You could have heterogeneous impacts on gene expression. How do you deconvolve that at the end model when you ask your question?
No,
because
we
if
don't.
You're pushing,
That's the whole point, actually. It's a little bit that we don't, because the way we see it is that then we have 1,000 clones. So like there's some variation of where it's put, like expression levels, all that sort of stuff. So you can see it as like, instead of having one cell line that is derived from one clone, we have all those different, so we just look at it at a population level rather than, cell line population level, right? Not real. So because we feel like that gives much higher, so this was done not by the core, but I did this when I was in the lab of Johan Jakobsson that works with transposable elements here in Lund University. And what we feel is that gives us much more, so much less noise, right? That we have, and also much less likely to find some sort of cell culture artifact or gene editing artifact, that we actually do keep them together. So we don't separate them at all. We even bring together two guides, right? Because that gives us a less noise situation.
Sure, but then doesn't it also complicate the differentiation process in terms of getting to, so for example, if you're editing in iPSCs and you have this heterogeneous or scaled impact on gene expression, for example, and then you differentiate towards NPCs, you could, within, I don't know, 10% of the cells, have a preference towards mesenchymal lineage, or it could shift the balance of what you'd see in that end differentiation, right?
Yeah, I mean, I think definitely if you have a knockdown that lies around 50% or something like that, it makes it much more difficult because then you don't know where you're at with every single cell, right? So then you know that they will be different. In ours, you know, when we take the bulk and if we do the cut and run, we can see that there is nothing. There is no mRNA left. There is no, you know, activation mark. So of course, it can be that a few percent of these cells still have it, but it's just so little that we don't see it as a. But what it definitely looks like is that like 90%, 99% is completely gone. Because otherwise we would still see, you know, like RNA peaks and stuff like that. So if you have like a 50% sort of thing, you don't know what the situation is. Is it 50% in all the cells, or is half the cells 100% knocked down and the other ones are wild type? That you don't know. So if you want to keep them as bulk, you really need to get up there with the knockdown efficiency, so that you know that you're dealing with the majority of them are the same, and they are basically have a really high efficiency. If you wanted more like dose it and those sort of things, then you could be better off like going with some sort of further selection.
Cool. Thank you very much, and thank you for a great talk.
Thank you.
So thank you very much. I think we do not have any other questions. So Pia, again, thank you very much. It was a pleasure having you as a speaker, and I hope to see you soon in Lund.
Yes.
The other, I think you are free to go for lunch, and see you tomorrow for the next round of conference. Pia, thank you very much.
Thank you. Very nice. Thank you.